Relationship testing in the blood bank. So we have parentage testing. So this tests genetic markers inherited to confirm or exclude biological relationships. This is now called relationship testing as it covers more than paternity, maternity. This can also include criminal cases, immigration, and identifying remains from disasters, wars, or genocide. So the history of parentage testing. So the ABO blood group test, first used to exclude non-paternity in Vienna back in 1926. Other blood groups and inheritance patterns were later applied. And then enzymes and proteins were started to be used for paternity testing around 1955. The next step in genetic testing, so in 1972, the Evian workshop demonstrated the extreme polymorphism of the human leukocyte antigen, or HLA system. The genetic system studied red blood cells, proteins, white blood cells, and it studied that it shared the reliance on biochemical expression of genes. These are collectively known as the classic genetic systems. Back in 1985, hypervariable mini-satellites called variable number tandem repeats were discovered. New genetic systems now directly assess DNA variability. These DNA-based polymorphisms are the current standard. So initially, VNTRs were tested using restriction enzymes on extracted DNA. This was followed by southern blotting and hybridization to variable number tandem repeat probes. This method is called restriction fragment length polymorphism testing. So the RFLP testing was eventually replaced by PCR technology, or polymerase chain reaction. So initially, PCR detected polymorphisms that were caused by point mutations or sequence-specific polymorphisms. PCR was then applied to analyze VNTR loci with small repeat units of about 4 to 5 nucleotides, known as short tandem repeats. So the primary objective is to confirm or refute a specific biological relationship, usually between a child and a parent. The aim is to exclude falsely accused individuals and provide strong evidence for inclusion if not excluded. Most laboratories have now transitioned to DNA polymorphism testing. The preferred method uses short tandem repeat systems. Polymerase chain reaction, or PCR technology, is still the standard for analysis. For red blood cell antigens, there are 6 blood groups that were routinely used in the paternity testing. Those include the ABO, the RH, the MNS, KEL, Duffy, and KID, though very few laboratories still conduct antigen testing for cases of question percentage, and that's due to the PCR testing that they currently do now. So the HLA complex, this is the most polymorphic genetic system in the human genome. It consists of multiple linked loci expressed as class 1 and class 2 antigens. In classic non-DNA parentage testing, only antigens from the HLA-A and HLA-B loci are tested. For interpretation of the classic testing results, so it is always assumed that the tested mother is the biological mother, and the mother and child are expected to share at least one allele. If the alleged father lacks the paternally inherited allele in the child, he is excluded as the biological father. If the alleged father is not excluded, a statistical analysis is then performed. This analysis determines the relative probability of the alleged father being the biological father compared to a random man unrelated to the mother or child. So most parentage tests are to establish paternity for children, often for child support. So for collection, storage, processing, and testing of samples must be carefully documented, and there's usually an unbroken chain of custody since this will be, for our court case or something like that. The tested individuals must be carefully verified to make sure that this is who they say they are, and any unauthorized access to samples or results is strictly prohibited. The AABB started accreditation, standardization, and quality practice efforts in parentage testing. The first edition of Standards for Parentage Testing Laboratories was published in 1990. An accreditation requirement manual was published in 1991, and then the CAP initiated a proficiency testing program for parentage testing labs in 1993.